Then in the first years, because the human body have internal magnetic fields influenced by external ones. If the external intensity higher than the internal could induce interferences and the molecular changes. Molecular effects as interferences can take place releasing metal ions from proteins for metalloproteins. The 80% of the proteins in our body are metalloproteins. In the next figure, figure 3, we continue. I don't see my possible. Yes. We continue with the same, with Y possible effects. The release of essential metal ions, enzymes, and proteins as iron from hemoglobin, transferrin, ferritin, iron, zinc, and copper from others, from metallothionein, copper from ceruloplasmin, calcium from osteoportin and calmodulin, and zinc is a cofactor that more than 1,000 enzymes. In the human body, there are billions of chemical reactions per second to obtain energy, which is delivered through the food we eat and liquid we drink. Whenever electrons flow in the conductor, they produce a magnetic field. Energy comes through the chemical reactions, and the electron flows come from the 3 main nutrients, named metabolism, while breaks down the carbohydrates, lipids, and proteins to provide chemical energy in form of ATP, adenosine 3-phosphate, and all the chemical reaction moving electrons and producing electromagnetic fields during these processes. Also through catabolism, glycolysis, and glucose beta oxidation, acid and citric acid, Krebs cycle, and electron transport chain, synthesis of ATP. All that occurred in the system of organs and activity essential metal heavy cabin metals demonstrate that overload, not yet, synthesis have metals influence time, conductivity and permittivity in electromagnetic fields. Electrons elsewhere are moving, and there are interferences occurring in electron transport chain, the mitochondrial membrane, etc. This interference can occur in numerous electron clusters. In the figure 4, we are all exposed to electromagnetic field from antennas, cell phone, booster antennas, etc. In the figure 5, we see the antenna 5G. We have proliferated today and is near in our house. In the figure 6, the possible interference between external and internal magnetic field, the external magnetic field, as now we have the frequencies in our magnetic spectrum, frequencies on the electromagnetic spectrum that we see now in the figure, from the left to the right, not yet, from the left to the right, is from gamma radiation entitled radionuclide, and to the right, X-ray, ultraviolet, infrared, and in the middle, we see the daylight with colors. The daylight and frequencies in Earth between 10 power to 14 and 10 power to 16. Hertz frequencies outward on the right, microwave, radio waves successively to low frequencies and extremely low frequencies of static fields. And in the next figure, yes, in the human body, electromagnetic fields are generated by brain activity transmitted with electric impulse from the brain to our hands and to our feet by chemical reaction and electro-moving and chemical reaction. Also important is the neuromuscular transmission during our exercise and blood flow. Numerous electron flows as electron transport chain, changing electrons as before mentioned. Electromagnetic fields are then the expression of our vital existence, which can be registered by the electrocardiogram. In the next figure, figure 9, as we see the electron transport chain schema from nicotinamine adenine dinucleotide, NADH, to the molecular oxygen, has the potential gradient difference of 1.1 volts. It is very great measure. It is justified the electron transfer through the chemical reaction as before mentioned. In the next figure, figure 10, the electron flows in the human body are influenced by external interference as higher than the internal fields, and there are the cause of the possible induction of disorders through different mechanisms of useless intensities. By different already mentioned mechanisms, the more important are in the mitochondrial electron transport chain and electrochemical central nervous system transmission, muscular contraction, cardiac electric myocyte transmission, pumping blood in our head, and encephalic electric impulse transmission, for instance, brain to our feet, also as before mentioned. In the next figure, figure 11, we show Antonius Galvani experiment in 18th century and how we move for magnetic or electric impulse, proceed to a electron movement, and finally to chemical reaction and movement. In the next figure, frequencies in electromagnetic fields exist at various frequencies, which are characteristic of their sources. Static fields, they are constant with time and direction and can be either electrostatic or magnetostatic. Their characteristic frequency, 0 Hz, time variation. Electrostatic fields are produced by charges at rest, and magnetostatic fields are produced by charges that move at the current speed. The strong higher of 1 Tesla magnetostatic fields are regularly used in magnetic resonance imaging instrumentation in clinical hospitals. In comparison, the Earth's magnetic field intensity is at its surface is 25 to 50 microteslas. And the domestic electrical appliance 0.02 to 7 microteslas. In the next figure, the figure 13, in the next figure, we continue with frequencies in electromagnetic fields as it deals with high non-applied frequencies. We go to the following figure 14. The next figure 14, some application, yes, some application of magnetic field in low on the left, low frequency, electric current used to fit appliances, induction heaters, and radio frequency and radio. In the radio frequency on the right hand, you see the radio frequency and television at about 88 megahertz. Afterward, heaters in-house, mobile phones, and bus stations, 900 megahertz, and microwave oven and dryers, also in our house, 2.45 gigahertz. Radar, 1 gigahertz to 300 gigahertz. In the next figure, figure 15, possible mechanism of action, electrostatic fields can produce forces on any charge, either at rest or moving. Magnetostatic fields can produce forces only on moving changes, deviating then direction, but cannot create new currents. And in low frequency fields can induce currents in any body that contains charges. Radio frequency fields can induce radiation on body has a penetration deep that decreases as the frequency increases. In the next figure, we show the mitochondria structure inside the cytoplasm of the cell with the intermediate space in out and inner space membrane supporting the electron transfer chain that we see in the next figure. The next figure, 70 figure, the physiological animation in the crest of the mitochondria, where the electron transport chain takes place. And in the next figure, figure 18, transport of charges to inside and outside the mitochondria membrane. Electron goes from NADH, as we have mentioned before, nicotinamine adenine dinucleotide, to the molecular oxygen through the enzymatic complexes and proteins outside. And in the next figure, the synthesis of ATP. No, the before next. The before. This one. The synthesis of ATP by the enzyme ATPase. And we see how ATP is synthesized to give energy for our body, intellectual energy and activity energy, exercise energy. And in the next figure, yeah, we see the cell and the mitochondria inside. Outer compartment to the mitochondrial is in green, and inner compartment in light blue. Are shown the enzymatic complexes of the oxidized phosphorylation, integral membrane of the mitochondrial, and also the Krebs cycle in the middle through proteins and the enzymes of the electron transport chain. And necessarily, it is synthesized ATP in the mitochondria for our energy. There are also the enzyme complexes all around the membrane. And in the next figure, material and methods, in figure 22, we applied extremely low frequency magnetic fields, 50 Hz. Determination enzymatic activities of the electron transport chain complexes in brain of bulb-shaped mice expresses to a magnetic field of 50 Hz, frequency 27.5 Gauss, during 1 to 4 weeks. Experiment as performed in extract of dissected cerebral regions, hypothalamus and thalamus of mice, and determined by spectrophotometrically through kinetic colorimetric reactions. And in the next slide, we see the calibration of the solenoid chamber, where there are inside the animals, the solenoid chamber of magnetic fields of 50 Hz. Magnetic field inside the chamber in Gauss and on the south, and position in centimeters. Magnetic fields inside the chamber in Gauss and intensity in amperes, as you see. And in the next figure, the exposure to magnetic fields of 50 Hz and 2.75 milliteslas and 27.5 Gauss. Male bulb C mice 1 to 4 weeks. On the figure, the seen solenoid camera installation after time of exposure 1 to 4 weeks. The following is kill the mouse, dissection of hypothalamus and thalamus, homogenization with buffer 1 to 3, centrifugation to 1470 RPM, that revolutions per minute, chemical reaction, and kinetic indivisible band of the spectrophotometer. And in the next figure, we see the result and discussion. The figure 26, how we show the chemical reaction realized, the more sensible the complex 3 and 4, because they are very long. They are the chemical reaction expressed in this figure. And we go to the next figure with explain the results. The result of the determination of the enzymatic activities. The complexes with higher activity were 3 and 4, as we have already said. Complex 3 is the coenzyme Q cytochrome C reductase, and complex 4 is the cytochrome C oxidase, is quantified by oxidation of cytochrome C reductase at 550 nanometers. And in the next figure, we see the enzymatic activities in hypothalamus and thalamus of exposed mice 50 Hz, the same as before. The graphic shows that specific activity depending on the days 7, 14, 21, 28 in the complex 1 and 4, 5, 1 and 5. We observed a greater enzymatic activity at 21 days, third week, and then habituation to the effects of electromagnetic fields because it diminishes a little the enzymatic activity. And in the next figure, figure 28, enzymatic activity referred to citrate synthase in the hypothalamus and thalamus expressed in this figure, greater enzymatic activity, habituation, yes. And the figure, this figure, the brain structures analysis analyzed till now showed the time effect relationship at the third week. We observed that the pair marked individual higher responses for the activities of the two cytochromes of the complexes 3 and 4 in comparison to the control animals. This fact could show the existence of the individual hypersensitivity due to physiological and biochemical conditions for certain animals because only few animals showed very high enzyme activities. In the next figure, figure 30, disturbances of the electron transport system possibly induced to installation of pathologies connected to activity and toxicity. But in scientific evidence, hypersensitivity under electromagnetic fields have not been till now diagnosed. And the figure 31, the next step is this one. After the Council of the European Parliament, it must be studied the simultaneous presence of electromagnetic fields and chemical agents in toxicology. It is the next step of our work we do in cellular structure and applying the dye neutral red who goes through the blood-brain barrier and show it permeability. The European Parliament in the official journal European Commission number C205 and document A3 specifically counseled to study the possible effect of the concurrence of chemical agents with electromagnetic fields in biomedicine. The simultaneous incidence of physical and chemical agents can induce to an unfavorable influence or unpleasant situation in humans. In the next figure, 32, some data on the exposure of rats with overload of mercury, plum, and cadmium microlevel 2.45 gigahertz. The next figure, 33, mineral elements applied, iron, cobalt, nickel, zinc, and a series of heavy metals, but there now we show the result applied only with lead. Elements administered in male Wistar rats by intraperitoneal route 1.1 milligram kilo day, except mercury is a little diminishes the dose. The elements with higher effects in conductivity and permittivity were heavy metals, lead, mercury, and cadmium, and iron. After 12 days,
लगेच दिलं तर टाकून कसं चालेल नुसतं बाळा पूर्वा
¿cómo? Ya me llevamos media hora, half an hour. Blood extracted rat organs were dissected, cast in 2 mm thickness, and measured the electric conductivity permittivity placed was measured, and in figure 34, the next figure, experimental system of the tissue exposure rat frequency magnetic field. That in the next figure, the strategy according to European Parliament and the scientific proposal chemical compound magnetic field nervous system, exposure to electromagnetic field of tissue animals with normal physiological condition, and transference of permeability to the membranes, brain, other systemic organs. And finally, determination of biological effects of the magnetic field in concurrence to environmental pollutants and heavy metals. A study of the synergy between magnetic fields with heavy metals, overload on the permeability of the blood-brain barrier and other physiological systems. In the next figure, figure 36, result and discussion, statistical analysis partially finished with conductivity and permittivity to some elements. And in the next figure, the extremely low frequency magnetic fields and heavy metals together induced to the permeability of the blood-brain barrier to neutral dye. Toxic metals have a higher action, and we discuss now, have an action than magnetic fields. Then the heavy metals are dangerous as the magnetic fields. Heavy metals alone, mercury, cadmium, and lead, induce a higher permeability to the blood-brain barrier than magnetic fields. Toxic metal together with electromagnetic fields in rat induces synergism, the passage of neutral red dye to the brain. And in the next figure, 38, the synergism in the permeability of the blood-brain barrier to neutral red dye. Heavy metal ions alone have an affinity to the enzymatic SH group, the thiolic group of the proteins. There are the mercury, plum, cadmium, and all other metals, essential metals also, bind to this SH group, the thiolic group. Electromagnetic fields could induce vibrations in the release of metallic ions for the enzymes and protein molecules bound to thiolic group. Electromagnetic fields also induce perturbation in the permeability of the cell membrane. In the next figure, we observe the electric properties of tissues, conductivity, and permittivity are modified by the overload of heavy metals. There are known human diseases in pathology with overload, that is osmosis, pneumoconiosis, siderosis, etc., for accumulation of heavy metals. In the next figure, 40, the figure number 40, the metal accumulation diseases are, we know, the Wilson disease, the copper, hemochromatosis, iron, siderosis, lead, hydrargyrin, mercury, parakeratosis in Japan, Itai disease, cadmium. And in the next figure, 41, we see the different hematological parameters, the values of the control and lead. In the last column, in the first column on the last range, is remarkable the increase of platelets, the electromagnetic fields which facilitates the formation of thrombi, like the COVID-19, the facilitation of thrombi, which is in the last range. And in figure 24 to 42, next figure, we see the values of the different biochemical parameters of the control and lead-injected animals. There are no remarkable value, only general inhibition is not important due to the toxicity of lead. In next figure, 43, we see the different parameter values in the control and enzymes. In the control lead and the enzymes, and we see the glutamic oxalate transaminase, GOT, important in the mitochondria, obtaining energy in the Krebs cycle. Also, the increase of lactate dehydrogenase in anaerobic glycolysis pathway catalyzing the reversible conversion of lactate pyruvate to pyruvate and vice versa. And also the NAD, the reduction NAD to NADH. And creatine phosphorkinase or creatine kinase, the regulation, the concentration of ATP, adenosine, it is the regulation of obtaining energy. Finally, it is shown the inhibition of the activity of the alkaline phosphatase. It's a zinc enzyme. As I mentioned before at the beginning of my speech, zinc is cofactor of more than 1,000 enzymes. Zinc is very important, is the cofactor of alkaline phosphatase. Alkaline phosphatase is very important enzyme in kidney, in the brain also, and in the bone. And is important, and is inhibited because zinc is replaced by lead, the toxic. And that is the cause, that's the diminishing, that is the inhibit the concentration of this enzyme. In the next figure, figure 44, we see the chemical concentration in ppm in red bars, and rats with cadmium overload and the control ones. And the next figure, we see the inhibition of conductivity of the rat tissues with lead overload, principally in kidney, also in other organs, but kidney is the excretion organ for us, and where the toxic go away, cadmium and lead and mercury and etc. And in the next figure, we can add the overload, mineral element by excess or ingestion of diet toxicology induced two generations of free radicals. Free radicals, they are toxic. Or radical oxygen and radicals of organic substance, also from our food. Similarly, the excess of toxic metal ions induced a plethora of electrons. And finally, in figure 41, next figure, in the last figure, we remember that the 80% of proteins are metalloproteins in the human body, and the essential trace element change can be healthy or toxic, can be activating enzymes in metabolism or be toxic. It depends on the concentration. Also be healthy, and some could develop some pathological diseases. Finally, there are no scientific data based in evidence medicine in low-frequency magnetic fields could have a pathological effect in humans, or that is important, or in hypersensitivity in immunodepressive patients. Thank you very much for your attention.
Hi, this is Ruchika. Thank you for giving such an informative presentation, Dr. Bartolome Ribas Ozonas. And yeah, if you have any questions, please post it in the chat, or you can unmute and ask directly from Dr. Bartolome.
If it is